Hello everyone, we'd like to get the presentation started. Welcome to our educational webinar titled 3D Modeling of Skeletal Muscle for In Vitro Functional Assaying. My name is Cora Lewis and I'm a product manager at STEMCELL Technologies. I will be your moderator for this webinar. It is now my pleasure to introduce you to our speaker today. Dr. Penney Gilbert is an associate professor at the University of Toronto in the Institute of Biomaterials and Biomedical Engineering. She holds cross-appointment in the Department of Biochemistry and in the Donnelly Center. Dr. Gilbert received her PhD from the University of Pennsylvania under the mentorship of Dr. Valerie Weaver, and then pursued postdoctoral studies with Dr. Helen Blau at Stanford University. Dr. Gilbert's team engineers and studies three-dimensional models of human skeletal muscle and explores muscle stem cell mechanobiology with the goal of identifying signaling pathways that can be tweaked to boost the function of skeletal muscle stem cells in the body. She is the recipient of an Ontario Early Researcher Award and holds a Tier 2 Canada Research Chair in Endogenous Repair. Let me now hand you over to Penney to begin the presentation.
Hello. It's really a pleasure to have an opportunity to share with you all a little bit about the research that's ongoing in my lab here at the University of Toronto in the area of 3D modeling human skeletal muscle tissue. Our overall research area of interest is adult skeletal muscle tissue. Now, this is a tissue that is found throughout your entire body. It allows you to do things like move and blink, to share expression with one another through our face, and perhaps most importantly, to breathe through our diaphragm muscle. One of the truly wonderful things about skeletal muscle tissue is that it has an inherent capacity to repair itself through the function of a small subpopulation of cells that sit inside skeletal muscle termed satellite stem cells due to their anatomical positioning. If you look at the top left image in this particular slide, you'll see an electron micrograph taken by Alexander Moreau in the early 1960s that's honed in and focused in on a single skeletal muscle fiber on top of which is a satellite cell. And what you'll notice about this cell type is that it has a huge nucleus and a very, very small cytoplasm, and it's sitting just juxtaposed to the muscle fiber. And it was proposed in this original paper that this cell population may be responsible for waking up and making lots of copies of itself when skeletal muscle was injured. And work since this time has shown that this population of cells can be marked by the transcription factor PAX7, and that when skeletal muscle tissue is injured, this population of cells in the adult is in fact required to repair the tissue in full. So just like many other tissues in the body, one of the aspects of skeletal muscle is that it declines in function and also in strength with age and with a number of genetic disorders. So things like Duchenne muscular dystrophy or aging, or even muscle atrophy that's caused by absence of gravity or bed rest. And so a lot of research in my lab and also that of many others throughout the world is focused on trying to understand how muscle stem cells are engaged to do their job and how we might develop therapeutics to improve the function of these cells through stem cell transplantation therapies, replacement tissue, or what my lab is interested in is systemically delivered drugs to boost the function of the stem cell population in the tissue. So we go about this task in a number of different ways, but one of the areas that my lab is very interested in and very focused on is in the development of 3D models to study healthy, injured, aged, and diseased human skeletal muscle tissue. We've developed devices and methodologies that allow us to look at a number of different aspects of human skeletal muscle biology in a dish. And today I'll tell you a little bit about two of those areas. First, our work in the area of modeling the human neuromuscular junction in a dish, and additional work that we've done in the area of scaling up our systems to be able to enable functional assays and compound testing to look at modulators of human skeletal muscle strength in culture. Most of our work in the area of generating 3D human skeletal muscle tissues begins in the following way. And I should say that all of this work was begun in my lab by a very talented PhD student, my second PhD student, Mohsen Afshar, who recently defended his PhD. So Mohsen received biopsy samples from our clinical collaborators at St. Michael's Hospital, Howard Ginsberg, Henry Ahn, through clinical collaborator Katie Tung. And with those biopsies, he would dissociate the tissue into cells, which he then would expand in culture to then derive muscle progenitor cell line and fibroblast-like cell lines that he would then mix together with a protein-rich scaffold and then seed into a device to allow the cells to reform tiny little human skeletal muscle tissues in a dish. Now, in order to have a system that allows you to support the self-organization of these human muscle cells to reform aligned multinucleated muscle fibers in a dish, these cells will require some structural cues. In the case of skeletal muscle tissue, this tissue typically is attached to bone through tendon, and this establishes uniaxial tension. And so our experience and that of others has been that any culture situation where you can recapitulate the activity of the tendon to allow the cells to establish uniaxial tension is sufficient to allow you to reform human microtissues in a dish. So this is one example of a system that we use to reform human skeletal muscle tissues in a dish. This is not a system that we developed. It was developed in the early 1980s by others, but it's a very simple approach and really highlights a minimalistic approach to reform human skeletal muscle tissues in a dish. Now, what you're looking at here on the left is a 12-well plate. And if you look down into each well, what you don't see is that there is optically clear rubber that has been cast into the bottom of each of the well as a liquid and allowed to harden. And then we place on top of that a white piece of plastic that we've laser etched into the shape of a dog bone, which is placed on top of the first layer of hardened clear rubber. And then we pour an additional layer of liquid rubber on top of this white dog bone and allow that to harden. And then upon removing the small little dog bone piece of plastic, what you're left with is a dog bone shaped depression in each of those 12 wells. By gluing a piece of Velcro or any sort of material with little hooks onto either side of this dog bone, you can now create little anchorage points where the muscle cells can attach and use those anchorage points to establish uniaxial tension. So in the red blown up area, what you can see is if you focus in on the center of this little well, you'll see a muscle tissue that has formed by self-organizing across the two black strips of Velcro on the top and the bottom. And if we take this image and we flip it to the side and immunostain, what you can see is many, many, many aligned multinucleated muscle fibers. You have your nuclei in blue, and you can see the beautiful striation of these multinucleated human muscle fibers. Now, just as others have seen, we can take these muscle tissues and we can stimulate them with electrical stimulation or chemical stimulation, in this case acetylcholine, and we see that these multinucleated striated muscle fibers are competent to contract. And so clearly one of the advantages of having a 3D culture system is that you can study one of the very fundamental properties of skeletal muscle tissue, and that's its ability to conduct force. But are there other questions that are very well suited to studying studying skeletal muscle in a 3D format as opposed to in a more simple system like a 2D culture dish? So we spent some time trying to compare the merits of studying human muscle progenitors as they fuse to form multinucleated structures in two-dimensional cultures, so petri dishes, versus in a three-dimensional system like the microtissue system that I just showed you. to understand what sorts of scenarios were best suited to studying a process in 3D versus studying in 2D. One of the things that we noticed very early on is that in our 3D cultures, we would see the formation of very uniform muscle fibers, whereas in our 2D cultures, we would find that over time, we lost our big fibers and we're left with smaller fibers. We could quantify that. So in this bar graph on the left, in the gray bars, you're looking at the diameter of the muscle fibers that are formed in 3D culture, whereas in the black bars, you're looking at the diameter of muscle fibers that are formed in 2D culture. And what you'll notice is that at 1 week of culture on the x-axis, the fatness of the muscle fibers looks pretty similar in 2D and 3D culture. However, already by 2 weeks, you can see that the size or the width of the fibers in 2D culture is starting to go down, whereas in 3D culture, it progressively goes up. And in our experience and that anecdotally of others, this really is simply explained by the fact that in 2D culture, you do have formation of nice muscle fibers, but as soon as they're competent to contract, they do contract and they break and they fall off of the dish, which then enriches for the smaller muscle fibers. Whereas in 3D, the 3D architecture can support the further maturation of those muscle fibers. So on the right, what you're looking at are Western blots of proteins that are associated with the maturation of the contractile apparatus in these human muscle fibers. And what you can hopefully appreciate is that over time in culture in 3D, we start to see emergence of the expression of adult forms of the myosin heavy chain proteins that are found in this contractile apparatus, such as myosin heavy chain fast and slow, whereas in our 2D cultures, predominantly we see expression of the embryonic forms, and we see very, very little expression of the adult forms compared to what we see in our 3D cultures. So in our 2D cultures, it isn't that we don't have expression, it's really just that the expression is so low compared to our 3D cultures that it's very hard to visualize it side by side on a Western blot. So from this comparison, we have a sense that our 3D cultures might be a nice setting to be able to study later stages of skeletal muscle fiber maturation, whereas a 2D culture dish is more than likely perfectly well suited to studying the proliferative phase of muscle regeneration and also those initial fusion steps and initial maturation of muscle fibers. Now, one of the other aspects that we noticed upon looking at our 3D cultures was that of the acetylcholine receptors. Now, acetylcholine receptors sit at the surface of muscle fibers, and as muscle fibers mature, these receptors cluster and end up being the contact point for motor neurons that connect and allow the brain to instruct the muscle to contract. And what we found in our 3D cultures was that over time, the size and the complexity of these clusters increased more so than in our 2D cultures. Now, in our 2D cultures, what we noticed was that while we did have clusters of acetylcholine receptor, they were very, very few. So if you look at the first graph just to the right of the immunostaining images, you'll notice that in the 2D cultures, you have very few acetylcholine clusters per muscle fiber when compared to those fibers that are grown in 3D culture at 2-week time point. Interestingly, when we looked at just the average size of the clusters, the average size of or area of the cluster wasn't terribly different in our 2D versus 3D cultures. But one of the things that we noticed was that in our 3D cultures, it was very clear that the complexity of those acetylcholine receptor clusters was quite distinct from our 2D cultures. And you can see that sort of nicely in the week 3 immunostaining image, where you can see a nice representative example of what appears to be a perforated structure that's sort of pretzel-like and reminiscent of what you might see in an adult skeletal muscle tissue, as opposed to what we would typically see in a 2D culture, which is represented by a week 1 image, where they're more plaque-like and rounded. And so by measuring, quantifying lacunarity, which is a metric of gappiness, and tells you a little bit about how morphologically complex these immunostained acetylcholine receptor clusters are, we can see in the far right graph that our acetylcholine receptor clusters in 3D are more complex than those in 2D. So with this in hand, it was starting to emerge that those human muscle fibers that were cultivated in the setting of a 3D culture system seemed to have a more mature contractile apparatus, and we started to see hints that the acetylcholine receptor at the surface of these fibers cultured in 3D might have matured a bit more than what we see in our 2D cultures. So with that observation in hand, we reached out to our collaborator, Randy Ashton at the University of Wisconsin, who at the time had developed a new protocol to push human-derived embryonic stem cells to the motor neuron lineage. And so Randy's group, very importantly, had developed a strategy at that time to be able to push the embryonic stem cells to an OLIG2 positive fate, where those cells were still happy to continue dividing a little bit. And you can see that in the bright field image of day 6. And he was able to take these cells and freeze them down and thaw them again and allow them to undergo a few more series of divisions. And this was important because that then allowed his team to send us cells in Toronto that we would then push to post-mitotic motor neuron lineage for the final 2 weeks of differentiation in our lab here in Canada. And what we would have at the end of this protocol were motor neuron clusters where you can see a ball of nuclei, and hopefully you can appreciate in the immunostaining image on the bottom right, and also the phase image just above that, neurites splaying out from the ball of nuclei. And it is these motor neuron clusters that we then worked to integrate into the 3D human skeletal muscle platform to then see if they could communicate with one another. Now, initially, we tried a number of approaches to coax these two cell types to grow together. We tried difficult things like producing 3D human skeletal muscle tissues and then placing very carefully a cluster of motor neurons on top of that 3D tissue and then hoping that it would regrow its neurites. But ultimately, what worked most reproducibly in our hands was something that Mosen tried one weekend, which was to simply, at the stage of making the human muscle cell slurry in the fibrin protein matrix, to just simply mix the motor neuron clusters together with the mononucleated human muscle cells and then seed that into the little dog bone device. And upon doing this, he found that these two cell types were quite good at self-organizing into their respective structures, and the motor neuron clusters, despite having been pulled off of the dish and having their neurites damaged, were really good at regrowing them back. And we can see that really nicely here in the next series of images. So what you're seeing here on the left is an image of that dog bone, where the top and the bottom black aspect are the Velcro strips, and outlined in red is that human muscle tissue. It has a sort of a denseness to it. And then if you look very closely, you can see a green rectangle that's highlighting a region of the tissue, which I'll zoom in on in the next image. So here what you can now see is the dense portion in the center is the muscle tissue. And you can see that just on either side of that muscle tissue, outlined in red, are two little bumps. And these are the motor neuron clusters that always like to make their way to the outside edge of these muscle tissues. Next, what we can see is the blue region that is highlighted in this image here. We will flip it to the side, and immunostain it, and hopefully you'll be able to appreciate that these two cell types self-organize themselves to form multinucleated muscle fibers with the motor neuron cluster on top and neurites making their way out and making contact with those multinucleated muscle fibers. The neurites are stained in red, and the muscle fibers you can see stained with sarcomeric alpha-actinin just below that. And if we zoom in just a little bit more, here we can see SMI32 labeled neurites, and they're tracking their way along these striated muscle fibers. And in white, we have stained the acetylcholine receptor clusters, and we can see that these neurites like to make their way to these acetylcholine receptor clusters just as they would in the body. One of the things that is quite striking is that if we focus our eyes in the areas in the center image that are outlined in the yellow boxes, we can see that these long neurites, where they tend to make contact, we see that the purple acetylcholine receptor cluster stain, bungarotoxin, shows up even brighter. And this is because everywhere that we see neurites making contact, we find that the acetylcholine receptor clusters are larger. And this makes sense because we know that neurite contact with muscle fibers is involved in the further maturation of acetylcholine receptor clustering. We can quantify this, and we can see that in the presence of motor neurons in the checkered bars, we have a greater number of acetylcholine receptor clusters per fiber in the left graph. In the middle graph, we can see that the size of those clusters increases in the presence of motor neuron clusters. And we also see that if we score for the complexity of those acetylcholine receptor clusters, that the complexity of the clusters is higher in the context of co-culture with motor neurons. It's all well and good that we see motor neuron neurites making contact with muscle fibers, but ultimately you want to be convinced that the nerves are in fact talking to the muscle fibers. And what we know is that usually the brain sends signals to motor neurons, which then lead to depositing acetylcholine vesicles into the neuromuscular junction, which then depolarizes the muscle fibers and causes calcium transients to form in those muscle fibers, and then induces the contraction of those muscle fibers. So to assess whether we have that process happening in our 3D neuromuscular co-cultures, we used two different approaches. We used a classical approach, electrophysiological recordings, in collaboration with two different groups here in Toronto, with Professor Mei Zhen and her postdoc Ben Mulcahy, and also with Professor Brian Stewart and his student Christine Nguyen. And with them, we were able to show that by stimulating the motor neurons with glutamate, that we then could cause depolarization of the muscle fibers. But what I wanted to show you here today is this video sequence. So what you see on the left is a 3D muscle tissue that is made from muscle cells that are stably expressing a GCAMP calcium reporter. And you'll see initially when we just add in buffer, HBSS, there's a little spontaneous activity, but it's not until we add in glutamate that now that you see all of the muscle fibers that are in close proximity to the motor neuron clusters, which are outlined in yellow, begin to show strong expression of this GCAMP calcium reporter. And of course, if we add acetylcholine into the culture bath at this point to saturate all of the innervated or even non-innervated acetylcholine receptors, we see a very, very, very strong calcium transient. And what's really striking about all of this is that these functional connections form in as early as 10 days in culture. And for those of you that have worked with 2D neuromuscular co-cultures, so human muscle fibers and motor neurons in 2D co-culture, you know that it's very challenging to see quite so many functionally innervated muscle fibers in 2D cultures at this time point, and really even at later time points as well. And one of the other things that you would hope to see if you're stimulating these cells with glutamate is a nice contraction of the muscle tissue, which hopefully you saw there. And we also know that that contraction is absolutely going through the neuromuscular junction, because if we add pre or post-synaptic blockers and then stimulate with glutamate, we see no contraction of the muscle tissue. And this is, of course, in contrast with what you saw initially, where if we add glutamate, we have a nice strong contraction of the muscle tissue. And so with this system in hand, it then allows us to ask the question, are there studies that are enabled by the availability of a neuromuscular junction culture model that has a really nice extensive innervation as the one that I've just shown you? And one of the aspects of neuromuscular junction maturation that's been very well known is the transition between the embryonic form of the acetylcholine receptor and the adult form of the acetylcholine receptor. So the acetylcholine receptor is an ion channel. It's embedded within the plasma membrane of muscle cells, and it's made up of 5 subunits. And during embryonic development, one of these subunits is a gamma subunit. And then as the organism matures, the gamma subunit is switched out for an epsilon subunit. And this process of subunit switching is very well known from studies of animals, but has been a process that has been very difficult to achieve de novo in culture. And for the most part, most people that might study epsilon subunit in culture will use enforced expression. And so we were keen to see whether by having such a nicely innervated muscle tissue, if perhaps we could extend the maturation of the acetylcholine receptor to now move from the embryonic to the adult form de novo. Now, one of the things that we know about the switch between the embryonic to the adult form is that neuregulin provided by motor neurons stimulates ERB receptors on the surface of muscle fibers to then induce expression of the epsilon subunit. And so we performed studies just to look at expression of the epsilon subunit on the y-axis in a number of different conditions. And so here, the black bars are 2D cultures of muscle fibers, and the gray bars are 3D cultures of muscle fibers. And you can see in the first row, we're looking at just muscle alone. And you can see, or sorry, I should say in the first column, we're looking at just muscle alone. And what you can see is that there's no expression of the epsilon subunit in 2D or 3D muscle cultures. If we add neuregulin to the culture bath to either 2D or 3D cultures, so this is the middle set of bars, you can see that you have a really dramatic upregulation of the expression of this epsilon subunit. And perhaps to our surprise, in our hands, we found that if we co-culture motor neurons together with 2D or 3D muscle cultures, we were able to see expression of the epsilon subunit in these 2 cultures. However, started to get a little interesting was when we looked at the level of the protein. So here you're in the top panels or the top row, we're looking at expression of the acetylcholine receptor epsilon protein, and the left column are 2D cultures without or with motor neurons, and the right column is 3D cultures without or with motor neurons. And hopefully you can appreciate that in both 2D and in 3D cultures, we do see expression of the epsilon subunit at the protein level, but it's only in the context of our 3D neuromuscular co-cultures where we see a stabilization of the protein, which then results in an increase in the protein that's available or being expressed in these co-cultures, which does not occur in our 2D co-cultures. So we then looked to see whether we could find any evidence that we we were getting functional integration of this epsilon subunit in our culture system. And again, we went back and we took advantage of our GCAMP calcium reporter expressing cells. And in this video, what you're going to see is first stimulation with glutamate to show you which muscle fibers are functionally innervated by neurites splaying out from the motor neuron cluster that in this video is outlined in yellow. And this will be followed by the next day a treatment with a peptide inhibitor that specifically binds to the epsilon subunit, and then subsequent stimulation with glutamate. And hopefully what you can appreciate is that we have a dampening of the signal, the calcium transients upon pretreatment with this subunit that blocks the epsilon subunit. So here we have our glutamate stimulation. And now in this series of videos, we've blocked the epsilon subunit and stimulated with glutamate. And you can see that many, but not all of the muscle fiber show dampened calcium transients, demonstrating that indeed in these 3D neuromuscular co-cultures, we have functional integration of this epsilon subunit into the system, which upon the various different experiments that we did in our 2D co-cultures, we were not able to find evidence of the epsilon subunit being functionally integrated into those muscle fibers. And so with this, we can conclude that a 3D culture model has many merits for studying the human neuromuscular junction. We see that skeletal muscle fiber alignment, maturation, and survival is improved in the context of the 3D culture. We find that in comparison to 2D culture, we have many functional neuromuscular junctions present within a 2-week period. And we also see that we have this de novo switch from the embryonic to the adult form of the neuromuscular junction, which is really advantageous in the context of studying disorders of the neuromuscular junction that are found in the adult, especially those that specifically target either through autoimmune or congenital ways the epsilon subunit. And so with this, the 3D culture systems seem to be a really nice way to study human skeletal muscle in a dish, but using the approach of a 12-well plate and casting each of these little dog bones each time can be a bit laborious. So a group of students in my lab asked the question, can we scale up this method and start to create something that's a little more uniform and that allows us to produce arrays of human muscle microtissues in a dish? And so the question that was asked by this group of students, again, Mohsen and 3 other colleagues in the lab, Sadi Davouti, Haben Abraha, and Mohammed Afshar, asked whether they could engineer miniature human skeletal muscle tissues in a dish that they would then use to screen for drugs that could alter human muscle strength. So to achieve this goal, they first set out to design a 96-well culture plate. And what you're looking at here is a near final version of this device, where on the left, you're looking at a top view and you're looking down, and you can see that you have a 96-well footprint. And then if we zoom in to one of the wells, as we're seeing in C, and look down, you can see that there's a circle, and in the center, there's an oval, and there are 2 little points looking up at you. And the oval is now the replacement for the dog bone channel, where we can see the human muscle cells in the fibrin scaffold across the 2 tendons, which in this case are 2 posts that you can see in D when we flip the well to the side, are pointed up, but then have a little hook on the side. And that hook is very important for ensuring that this contractile tissue doesn't pop off of the posts when it becomes mature. Now, what's really lovely about what this team of students did was they designed this, they created this design, and then they produced a hard 3D printed plastic positive, which they could then use to, through a series of casting steps, produce a hard polyurethane negative, And that negative can be used an indefinite number of times to produce an indefinite number of soft PDMS plates. PDMS is a soft rubber, and what we can see here, if we look at an example of this plate from the side, is we can see that this rubber plate is optically clear, which allows us to image through it. It's made of rubber, so it means that we can cut it. So if we want to do produce 96 tissues, we can, but in most cases we may want to use fewer, so we can cut it away and save the rest of the dish for another time. But perhaps more importantly, hopefully you can appreciate that in this single casting step, all of the features needed to make each of these little tissues is cast in a single step. So as opposed to having to glue little posts at the bottom of the well, the little posts are cast directly into each of the 96 wells. And this in turn then allows us, if in the right image, to produce arrays of little human muscle tissues in a dish. And so what that process looks like is this here. You can see in the bright field images on the top, at day 0, we're looking down into the well, and you're seeing a portion of that little oval, and you see that the cell slurry is spread evenly inside that little oval, and you can see little hints of the rubber posts on either side of the image. And even within 2 hours, the cells will pull on the extracellular matrix and establish uniaxial tension and align themselves along that axis of tension and fuse to form multinucleated fibers. And this causes the tissue to compact over time from day 1 to day 14, such that you have a dense tissue across the 2 posts. And then by 2 weeks, if we look at the bottom image, you can see here stained with sarcomeric alpha-actinin to visualize the contractile apparatus of each of these multinucleated muscle fibers. You can see that we have aligned multinucleated muscle fibers in these little miniature tissues, and the dark regions on either side of the tissue is where those posts were. And the advantage of having a plate that's completely made of rubber is that then if we stimulate these tissues to contract, as you're seeing in this video, you can see that the muscle tissue in the very center will deflect the rubber posts that acted as the tendons to allow them to form. And that in and of itself is very, very useful, because that deflection can be used to quantify the strength of each of these tissues. Now, the way that we do that is to first calibrate the system. What you're looking at on the left is one of those rubber posts that's being pushed by a hard metal rod. The little circle is a metal rod that's going down into the video, and the little hooked rubber post is moving up and down with the force of that metal rod. And that metal rod applies force at a known amount of force, and we can then correlate the amount of force needed to produce a certain amount of displacement, which then now allows us to quantify the strength of each of these tissues downstream of various treatments. And Haben, one of the members of this team, decided that it would be useful to have a semi-automated algorithm that allowed us to automatically detect and quantify post-deflection. And so what you're looking at here is the end result of his algorithm, where you can see a view of the rubber post from the top, and you can see that the algorithm automatically finds the edge of the post and then can track the movement of the post, which allows us to quantify the pixel of movement of this post in response to tissue contraction. So with these tools in hand, we're then in a position to be able to look at different scenarios where human skeletal muscle microtissues might change in terms of becoming weaker or stronger over time. And what you're looking at here is just a proof of concept. So you can see that if we look at post deflection of each of our tissues at different time points of differentiation. So at day 7 versus after 7 days of after 10 days of differentiation versus 2 weeks of differentiation, we can see that the relative force of these tissues increases with time, exactly as we would expect. But ultimately, if you want to show that your system is robust and that there's some value to your system above and beyond a classic approach that one might do in their own lab, you need to demonstrate a value out of your system. And so what we decided to do was to test a variety of different drugs and proteins that are known to increase or decrease muscle strength, and provide side-by-side comparison of a standard histological approach, so an endpoint assay, to assess the effect of those treatments on the 3D muscle tissues, as opposed to our non-invasive force measurement, which can be performed at many time points over time, so it can be used in a longitudinal fashion. So what you're looking at in these images on the left are muscle tissues that are immunostained for sarcomeric alpha-actinin at the end of a 2-week culture where the tissues have been treated with either dexamethasone in the top row, cerevastatin in the middle row, or insulin growth factor in the bottom row. The top 2 rows are drugs that are known to weaken skeletal muscle, whereas the bottom row, insulin growth factor, is a protein that's been shown in mice to increase muscle strength. And what you can see is that if you apply for 1 week different doses of the 2 drugs that are known to be myotoxic, at the end of your assay, or at your greatest concentration of drug, you can very, very clearly see morphologically that the multinucleated muscle fibers look more disorganized, they're less aligned, and they clearly are much thinner than the muscle fibers that we see in the control condition, so 0 treatment of drug. And we can quantify that in the graphs, and you can clearly see that in a dose-dependent way, as we increase the dose of these drugs, the muscle fibers' thickness or diameter goes down. So interestingly, if we look at insulin growth factor, again using this very classic metric to study muscle fiber, or I guess as a proxy of muscle fiber strength, so muscle fiber diameter, we can see that although insulin growth factor has been shown in animals to increase muscle strength, that in these cultures, even at our highest dose, we really do not see any difference in the width of the muscle fibers that we see in our culture. This is in contrast to when we perform instead our non-invasive assay to look at the strength of the tissues. And hopefully you can see here that in the control condition, we're looking at the movement of the post. The movement of the post is really minimal compared to what we see in our IGF-1 treated condition, where you can see that the post moves really, really dramatically, so, showing that through this non-invasive assay of function, we have a more sensitive approach to be able to assess treatment effect on microtissue strength. And we can see that here, where on the bottom, we're quantifying relative force as post-deflection for each of our treatments. In the first column, dexamethasone, and then cerevastatin, and then IGF in the far right column. And you can very nicely see that in all cases, with the 2 myotoxic drugs, we see a decrease in the relative force, whereas with our IGF-1 treatment, we can see an increase in force generation. And so this is nice because it then enables us to perform longitudinal analyses and just gives a lot more flexibility for those that are performing the assays. And so I'll just end there and give you a final snapshot of our tissue engineering team. and point out Mohsen in the bottom left, who began all of this work in my lab just over 5 years ago. You may notice on the top that his parents were very kind to clone him and then send him to the lab. His brother Mohammed is continuing the work that Mohsen started in the area of studying metabolism and exercise. And we have an array of other postdocs and graduate students who are developing assays to be able to study various different aspects of human skeletal muscle physiology and function in a dish. And I'll end there just by acknowledging all of our collaborators on the work that I've shared with you today and our funding agencies, and invite you to follow us on Twitter and to also check out our website. And happy to take questions.
Thank you very much, Penny. That concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. Let's start with our first question. When working with primary skeletal muscle progenitors, do you observe a donor age effect on the capacity for the progenitors to form myotubes and subsequently hypertrophy when cultured in 3D?
That's a really great question, and it's one that, surprising to us, we find that we really have not seen any obvious difference in terms of human muscle progenitors from young versus aged donors being able to make muscle fibers in our 3D cultures. And we think that that's probably because the way that you make a primary human muscle cell line is that you dissociate the muscle tissue into the mononucleated cell population, and you're relying on the muscle stem cell compartment to then expand and survive to produce the muscle cell line. And ultimately, you're going to enrich for a subpopulation of cells that are competent to do that. So presumably in your aged human skeletal muscle tissue, we know from a lot of work that there's a large population of the muscle stem cells that are less potent, but we also know that there are cells in there that are still good to go in terms of doing their job. And it's almost certainly that subpopulation that is being enriched and is available in our muscle progenitor cell line that we're using to make 3D tissues. So I think ultimately at the level of the muscle progenitor, at least in the readouts that we're looking at, we don't see any obvious differences between tissues made from young versus old donors. But as we expand the metrics that we assay in the 3D system, I imagine we'll start to see differences emerge when we compare those two scenarios.
Do you see any evidence of PAX7 positive cells in your 3D muscle structures?
So absolutely. We know in the field that there's a subpopulation of reserve cells that express PAX7 at a high level. And when we make 3D human muscle tissues and then immunostain for PAX7, if we hunt for a very, very long time, we can stumble upon a handful of PAX7 positive cells that do like to find their way to sit on top of the multinucleated fibers, although we don't see evidence of them forming their own basal lamina and being covered with a basal lamina as you see in vivo, but they do seem to associate with the muscle fibers in the tissues. So yes.
Are you planning to co-culture motor neurons with muscle cells in these miniature soft PDMS plates? If so, how are you planning to quantify the NMJ characteristics?
So we are at the very early stages of co-culturing motor neuron clusters with the muscle tissues in the miniaturized platform. And in this case, it will be key to really streamline the size of the motor neuron clusters to make sure that there's uniformity, and also streamline the number of motor neuron clusters that we add to each of the cultures. Because if we're going to start to do quantitative studies, then we need to start out with a system that's really nicely controlled. So we're working on methods to be able to uniform that part of the process first, because also ultimately we do see a bit more more spontaneous activity in our neuromuscular co-cultures. And in the context of this miniaturized platform, we want to make sure that we're not inducing too much contraction. And then ultimately in the 96-well platform, it's more challenging to do single fiber analyses than a larger format. A larger format is absolutely needed to perform things like electrophysiological recordings. So in the miniaturized format, our readouts would need to be things that are amenable to bulk analysis. So these tend to be colorimetric assays and the like, but we can image through the rubber platform. And so that does provide an opportunity to potentially visualize reporters and various different genetic reporters that become available. And so at that point, when there are more tools available, the 96-well platform becomes a little bit more interesting in terms of looking at single neuromuscular junction maturation or response to treatments. But for now, we're a little limited to global changes in neuromuscular junction activity in the 96-well platform.
Have you observed any shifts in myofiber subtype, that is fast versus low twitch myofibers, with your in vitro training of 3D muscle cultures?
So I didn't present our work looking at how contraction alters the maturation process or endpoint of our muscle tissues, but that is an area that we are studying. And at this point, it does appear that we do see some shifts in fiber subtype, but these are sort of early days. And ultimately, what we need to do is we need to characterize at a better level how the location of the muscle biopsy that we receive influences the ultimate fiber subtype of the tissues that we produce. At this point, we predominantly receive muscle tissue from back muscle from patients' dental surgery. And in order for us to have a better sense of the full range of the relationship between muscle tissues that are predominantly fast versus slow and how that manifests in our 3D system, we need to have access to far more muscle tissue from different regions in the body. And that will also give us more confidence about the interesting things that we're seeing in response to contraction. But I would say that for now, these are early days to draw conclusions.
Have you ever established 3D culture using human pluripotent stem cells derived from muscular dystrophy patients? And if so, what do you see?
So for our work, we initially started with primary muscle cells just because the protocols to derive muscle cells from pluripotent sources are still somewhat new. They take a very, very long time. And ultimately, the consensus in the field is that they yield muscle fibers that are relatively immature. And given the early state of the field of 3D human skeletal muscle modeling, it made sense to start with a cell population that we already knew was really good at making muscle and that was coming from an adult source. Having said that, we are now starting to collaborate with groups who make pluripotent muscle progenitors from pluripotent stem cell sources to then integrate them into our various platforms and use those for our studies to look at specific disease phenotypes. We have done work in the area of modeling Duchenne muscular dystrophy, and that work has focused on using primary muscle lines from Duchenne muscular dystrophy patients, and also taking advantage of resources developed by the AFM telethon to produce a number of immortalized human cell lines from patients with Duchenne muscular dystrophy and having paired control healthy samples to go along with them. And without stealing the thunder of the postdoc that's working on this project, I can say that we see many, if not most, of the hallmarks of Duchenne muscular dystrophy that you would normally report if you were studying mouse models, and even some of the hallmarks that you don't see in mouse models that are well known to manifest in humans. So I would say that this system is very powerful with regards to its capacity to study Duchenne muscular dystrophy, and also as a potential platform for preclinical assessment of therapies that impact several of the clinical features of the disease.
All right, and we have time for one more question. Have you ever observed any utrophin expression at the neuromuscular junction using your 3D model?
You know, we have not looked at that, but it is something that we definitely can easily do. And thank you for the suggestion. We'll make sure that the next time we set up a co-culture, we perform a staining.
Well, okay. I think that's all the time we have for today. I really want to thank everyone for attending, and I'd especially like to thank you, Penny, for your time and an excellent presentation. Thanks again and goodbye.